Welcome to the video on the Lifelines webinar Access to the Genetic Data. In this video we will be presenting the genetic data that we have to offer at Lifelines, and we'll show you the available data services that you can use to access this type of data. This presentation will be given by me, Bolmer, and my colleague Sylvia Gerritsma. We're both data managers at Lifelines. In this presentation we'll shortly look at the Lifelines dataset in general and give a brief introduction. Then we'll move on to the data services that you can use to access this type of data, including the genetic data. And we'll talk about the access procedure. And then we'll move on to the genetic data, in which my colleague Sylvia will show you some highlights, for example on the genotypic data that we have, but also the microbiome data. And then we'll conclude our presentation with 3 take-home messages. At Lifelines, we're collecting data and samples from nearly 170,000 participants, and our mission is to make this data available to researchers worldwide that work in the field of healthy aging. Some of our characteristics of our cohort are, like I mentioned, that we have nearly 170,000 participants, which is around 10% of the northern population of the Netherlands. We also have a lot of diversity in the collected information and the data that we have, of which the genetic data that we're discussing today is an example. We also have a very long follow-up time for these participants of at least 30 years. And we've now been collecting data for 13 years, so we're nearly halfway at the halfway mark. We also have a flexibility in our data collection, and we allow researchers to include additional studies in which they can propose, for example, their own questionnaire that they would like to send to participants. Our cohort is population-based and started with a healthy inclusion, but over time, of course, individuals can and may develop diseases. So you will also see that within the cohort. And it's based on a 3-generation design, in which we initially invited participants through their GP, and then, when they've been included, asked the participants if they had any family members or relatives that also wanted to join the study. And the Lifelines data is open access and available for any researchers on the topic of healthy aging. I would like to mention that we do not do our research ourselves, but that this is mainly done by other researchers from, for example, universities or university hospitals. So at Lifelines we have the complete range of data, ranging from the phenotypic data, which we ask a lot of topics in questionnaires to our participants, for example on lifestyle and environment, which you also see on the right side in this image, but also on education, disease development, and general well-being. We also have the genomics. That's something that we'll be discussing today and we'll be showing to you in this presentation. And besides the self-reported data, we also have environmental data from, for example, external data sources. The available data and some examples of the data are, for example, in regard to the phenotypic data, the measurements that we do. People also visit our locations in which we measure the blood pressure, we do an electrocardiogram, we test for cognition, lung function. Of course, we also have the questionnaires that we've sent on a regular interval. These are sent out whenever participants come at a location, but also in an interval of 1.5 years. And of course the biological samples, such as the blood collection, which is also done on location, and for example urine. And besides the phenotypic data, we have the genotypic data, which ranges from the genomics to metagenomics, transcriptomics, and microbiomics, to name a few. Well, this data collection ranges over a period of time. Like I mentioned, we've been collecting the data for 13 years now. We started in 2006-2007 with our baseline assessment, in which we sent out a first questionnaire, but we also had 2 visits. One visit for the measurements and one for the biological sample collection. Then we followed up with a questionnaire 1B and 1C on a regular interval, which eventually led to the second assessment, in which participants again came to our location for measurements. And now we're currently in our third assessment round, in which we're again inviting participants to come to our location for the measurements in the biological samples. And this will continue till 2023. Then, moving on to the data services. At Lifelines we have 2 data services available for you to use as a researcher to work on the data. One is the Lifelines workspace, which supports a Windows environment, and the other is the Lifelines high performance cluster. The first one, the Windows environment, is mainly used for phenotypic analyses. It's a ready for use solution, it's intuitive, and it supports applications such as SPSS and Stata. So if your research mainly focuses on phenotypic data analyses, this is definitely a service that fits your needs. The other one is the high performance cluster, which is facilitated together with our university hospital, the UMCG. This service is based on Linux and can be used for complex resource intensive analyses. And on this service we also host all of the genetic datasets. And this environment is more suitable when you need to do heavy computational analyses, or if you require a lot of data to be processed. So this is an example of the workspace. This is a virtual environment, so it can be accessed from anywhere, from any PC. And as you can see, it's a regular Windows 10 virtual environment, which has, for example, Excel, SPSS, and in the background you also see the folder structure and other applications. Then we also have the high performance cluster, and this is a screenshot of that service. And as you can see, this is a command line interface. It has no graphical support, so you cannot launch any applications such as Excel or SPSS. So this also requires a different set of skills to be used effectively. And that is also how it compares to the workspace. So a different set of tools and applications. It requires technical know-how to be used effectively and correctly. And as a researcher you also have more responsibility to execute proper data management. When you've gained access to the cluster for your genetic data analyses, you'll be able to access a shareable project folder. This might be useful if you're working with multiple researchers on the same project. You'll receive access to the requested genetic datasets, of which we'll show some examples later on. And you can access also this environment from any machine. This is an example from the cluster in which we showcase our datasets. So these are all examples of datasets that we make available through the cluster. And here you can already see some of the examples we'll also explain later on by Sylvia. For example, the cytosnip and the GSA data. Those are our largest datasets with the most amount of participants genotyped. But also, for example, the deep datasets containing the microbiome data and the methylation data. And of course also the phenotypic data, which you can use to combine with the genetic data. Well, these are some examples of genetic research that's being done by researchers from other universities. And this was all done with Lifelines data, and here you can see the variety of types of research that can be performed. So this is also something you can think about when you want to submit an application for Lifelines data. So the first one, for example, combined cardiovascular disease with genetics and gut microbiome. Another one was about human reproductive behavior and genetics. Third one was already with the COVID data that we've collected over the past year in combination with genetics. And of course also health and lifestyle, and for example nutrition and diet are very interesting topics to combine with on one hand the genetic data, but also of course the microbiome data. Which brings us to an important question: how can you as a researcher gain access to this data for your research? Well, the first step in this process is to apply for the data. In this case, for example, the phenotypic and the genetic data. You can do so by going to our website and filling in a form, but you can also of course always get in touch with us to ask about the possibilities and how it fits your research needs. Then after you've applied for the data. We'll review it and see if it fits the healthy aging theme, and if we can also provide you with the data you've requested for your research. After that, we'll send an offer and a DMTA, which you can sign, and after the signatures have been set, we'll be able to release the data and the samples to you, for example through the Lifelines workspace, or if your research is more based on genetic analyses, through the Lifelines high performance cluster, which will allow you to do your research and in the end come with the results in the publications, of course, in which you can always also get in touch with us again to see how we can share those results for you as well, and to also provide you with some more publicity. And now I would like to give the floor to Sylvia, who will be showcasing some of the genetic datasets that we have available, which you can use for your research.
Thank you, Bas. I will now give an overview on all the available genomic data at Lifelines. Here you can see a pyramid that summarizes which data was generated based on the Lifelines cohort. At the bottom of the pyramid, you can see the sample size of the Lifelines cohort, so 167,000 participants, of which we have approximately 2,000 phenotypes available. For about 36,000 participants, genotype data is available, which was generated using the global screening array. This is actually the second population which was genotyped. An earlier population of about 15,000 participants was genotyped using the cytosnip array. A whole genome sequencing was performed on 165 Lifelines participants, which are part of the GoNL project. This is a project on genome sequencing of so-called trios, so the mother, father, and the child. We do not have this data available at Lifelines, but it is available at GoNL. The microbiome was determined for a small part of the cytosnip array population in an initial study called Lifelines Deep. In the study on the same participants, also methylation profiling was performed, as well as RNA-seq and RNA sequencing and metabolomics. The microbiome was also studied for a larger population from the global screening array sample and for a number of participants in GoNL. In the next slides, I will go into more detail about these studies. In 2013, the first Lifelines population was genotyped. This population consists of 15,400 participants, from which the samples were genotyped using the cytosnip array. These participants were mostly unrelated, and they are all adults, which means they were aged 18 years and older. We have the genotyped samples after quality checks, QC, available, but no principal components, PCs. And the data was imputed using the 1000G reference panel. And last year, the data was also imputed using the haplotype reference consortium. And we have these different file formats available. The genotyping of the largest and most recent population was initiated by the UMCG Genetics Lifelines Initiative, or abbreviated UGLI. This UGLI consortium was founded by Harold Snieder and Siska Wijminga of the University Hospital of Groningen. And they brought together over 25 PIs who were interested in genetic research in Lifelines. The aim of the UGLI consortium is to genotype the whole population of Lifelines, so all the 167,000 participants. Last year, in January, the data of 36,000 samples became available, and currently another 30,000 samples are being genotyped using the Axiom chip. Later this year, another 30,000 samples will also be genotyped. leaving us with a population of 96,000 genotype samples within the ugly. So how were the samples selected within the ugly population? There's little to no overlap with the previous genotype population. So this is the population of 15,000 participants using the cytosnip array, apart from some samples which were used for quality checks. Samples were chosen from the largest families within Lifelines, and these were informative samples as the oldest and youngest generation of these families were chosen to be genotyped on the array. So all the other family members could be imputed using the data from the genotyped individuals on the array. This has not been done yet, but will become available in the near future. Like I mentioned, the global screening array was used to initially genotype 36,339 samples, which includes approximately 3,000 children. We have the raw data available, the genotype data after quality checks, and for this data, scripts, logs, and PZs are available. The data was imputed using the haplotype reference consortium, and we have this different file format available on the cluster. In one of the first additional studies performed with Lifelines data, the microbiome was analyzed for approximately 1,200 participants using metagenomic shotgun sequencing. 16S sequences are available for approximately 1,000 samples. We have raw data available from these two samples, and there's also overlap between these samples. In the near future, we will also make microbiome data available for 9,000 additional samples, and these samples come from the global screening array population, so the ugly population. The first microbiome study was performed by researchers from the additional study called DEEP. It is called DEEP since many analyses were performed on samples from this DEEP cohort, thus on the participants that participated in this study. The DEEP samples were genotyped using the cytosnip array, so SNPs are available. Methylation profiling was performed on approximately 800 of these DEEP samples or DEEP participants. We have RNA-seq data available, and metabolomics were analyzed. So we have metabolites in exhaled hair and blood available. On the right side, you can see a table where you can see the sample sizes of the different analyses which we have available. So we have a wide range of genotypic data available, which can be combined with all the phenotypic data that we have available on our participants. To access the data, you need access to the high performance cluster of the University Hospital of Groningen. And since last year, already 50 projects gained access to the data and are now performing their analyses. New genotyping projects are ongoing, so if you want to discuss the possibilities for your project, please get in touch with us. If you still have questions, please check out our wiki page, or you can email your questions to the Lifelines data managers at this email address. Thank you for listening.